as I mentioned, today's speaker will be telling us about deep learning for image segmentation. And Anna has a background in mathematics and worked as a software developer and scientific programmer at CERN in the Helmholtz Center before doing a PhD at the University of Heidelberg. From there, she worked as a visiting scientist at the HMI Genelia Research Campus before joining EMBL in 2018 as a group leader, where her group now works on machine learning for bioimage analysis. And we're really excited to hear about your work. Please take it away, Anna.
Thank you very much, Johanna, for the kind introduction and also for inviting me to come here and tell you a little bit about our research and the tools that we are building. Let me then try to share the screen maybe. And do you see anything?
Yes, we see your screen.
Very good. Okay, yeah. So I want to tell you about how we use deep learning for 3D image segmentation. And it's actually not just deep learning, but it's a whole little sequence of algorithms in a way. And I'll show you all of them, and I'll show you how you can use them yourself. Okay, no, too fast. Sorry, Zoom is somehow strangely responsive. Okay, so the particular problem that I want to tell you about is not just 3D segmentation in general, but the so-called boundary-based 3D segmentation. So this is a problem that you have in many imaging modalities. It's especially pronounced in EM, where if you want to segment cells or organelles that have a membrane, you have to start from a boundary and then build it up from there. But you can also have it in other label-free imaging, like in phase contrast, but also in fluorescence imaging when you want to study the cell morphology. And then you usually do that starting from some kind of a membrane marker. And then, so you have 3 examples here. In this talk, I will mostly be talking about the kind of data that you see on the right. So really when you have light microscopy and then in light microscopy, you have membranes labeled and you want to segment cells. And here I will tell you about the project that we have developed together with our collaborators who are interested in this kind of data. So they want to study morphogenesis of plants. And they have very many different plant body parts. And they want to segment them all and see how they develop. So here, this work that I'm going to show has mostly been performed by the 2 fantastic graduate students, Adrian Volny from my lab, and then Lorenzo Cirone from Fred Humprecht's lab at the University of Heidelberg. And so that's the kind of data we are concerned with. So this is the lateral root of the Arabidopsis. And you see it's actually growing sideways, but I have it up here just so that it would fit into the white screen. And we want to extract individual cells out of this. So the general approach that we take is we go from the boundary segmentation. And then, so we have a neural network, which is where the deep learning part comes in. So we have the neural network that predicts where the boundaries are. And then out of these boundaries, we then basically break it up into little pieces, which we then call superpixels. And then we agglomerate the superpixels into the final objects. And here you can also see the 2 data sets that I'll be showing throughout the talk, because these are our, so to say, workhorse data sets. And one of them is easy, that's the top one, right? So this is the oval of the Arabidopsis. And it's fixed sample, fixed cleared, image by confocal, very nice, extremely good contrast, really super good. And then we have the other sample, which is live imaging of the growing root with light sheet microscope. And that one is much, much more challenging. And basically when developing this whole pipeline, we always try to make it work for both of them and to have a method that would work not only on the easy data, but also on the difficult one. Okay, so where do we go from here? Right, so we start from the boundary prediction, and the boundary prediction in this case, we do with a 3D unit, and we predict short and long range affinities between the pixels. And so I have it here actually shown on an EM example, because when we work with EM data, we use pretty much the same pipeline anyhow. Right? But so in more detail, the UNet is a very old network. Right? It's been introduced by Ronneberger in 2015. Right? In the world of deep learning, this is really prehistoric time. Right? It's dinosaurs. Like nothing else from that time really survives. I mean, the ideas, of course, survive. But the networks, the particular implementations, the UNet is really remarkable in that sense. So people have been using it for a long time and tried to get the extensions of it to work, and then try to get other networks to work just so that you can show some novelty in a paper. We have also tried, and we have tried a lot of variants. And well, we still came back to the standard one because in the end, for us, that was always the best. Right? So if you want to evaluate on many data sets like we did, and you want it to work in a versatile way, the normal classic 3D unit was still the winner. So if you want to see the most popular variants, residual unit doesn't preserve the details as well. Dense unit, it converges super fast, right? It's a very nice network for like fine tuning. It's really, really fast, but it overfits. So I would say it converges too fast. We tried many other variants. They didn't bring anything either, right? We tried, combining the layers in different ways and different normalizations and this and that. But in the end, we kind of went back to the classics. So we have a 3D unit. And then, since I'm not trying to kind of publish this work again, I can freely admit that there is no novelty in the neural network there. And we predict the affinities between the pixels and not just if the pixel belongs to a boundary or not. And what the affinities are, you can kind of see in this illustration down there. So you see the affinity for the red pixel. To compute these affinities, what you do is you look at the pixel, to the one step in X, one step in Y, one step in Z. And then what you predict is, are they still in the same object? And then you can do that not just for one pixel offset, but for a few, which in case your boundaries are not very well preserved, which is almost always in microscopy, that can be much more robust than predicting for a single pixel if it actually belongs to a boundary. And this becomes especially important in anisotropic volumes, and especially in very anisotropic volumes, like if you work on EM data, and then you have thick slices GEM data, and you really need to work in affinities, because otherwise you have this like slices of sausage that lie on top of each other, and then they don't have a boundary between them, but they're still different objects. So you need this kind of representation to actually work with this data. Okay, but for simpler light microscopy data, nice isotropic, everything is continuous, you can also just predict foreground background. Okay, so standard unit. Yeah, some comparisons. So we ran a whole lot, and in the paper that we published about the plant segmentation work, we evaluate all of them, and there are like whole pages of tables comparing different variants. So you can see that the easy data, that would be pane A, and the more difficult data, that would be pane B. So you see that actually it's reverse, right? The red and the purple are the best for the difficult case, but not as good for the easy case. The ones in the middle are kind of okay for both. The difference there is not particularly significant, right? So it is significant. It's something where you can say, okay, that one is really better. But in your life in practice, when you will be using that to actually segment things and then do something with them, it's a difference on the level of correcting a few objects more. Okay, so in the end, what is our best CNN? It's a 3D unit, right? In terms of the loss, it's a binary cross entropy plus dice, like a linear combination. So dice, the dice loss here, if you're into neural network training, just wanted to note that. The dice loss here kind of compensates for the very unbalanced classes, because only a few pixels are actually membranes and most is background. Yeah, we tried small patches, big patches, these small patches with batch norm work the best. What is really, really important is to use a lot of data augmentation, and especially to use what we call the noise augmentation. So when you have your training data and then you corrupt it with very many different kinds of noise, right, and do it randomly a lot of times. So actually this data augmentation, especially the noise augmentations, I would say make a bigger difference than whether you take smaller or bigger patches and this kind of stuff. So yeah, so for the deep learning part, and I think for the CNN in general, that was our main message. So the 3D unit still works very well. This combined loss is quite stable. And with a lot of data augmentation and with sprinkling a lot of noise on top of your data, you can actually get pretty decent predictions. Right? So you can see that for our difficult data set, this is how it looks. Right? So if you remember how bad the raw data was, you would appreciate it's not so bad as a prediction. On the other hand, of course, it's also not perfect. And I hope that this image, so I stopped at kind of the worst slice, the rest are not so bad. And I did that so that you would see the motivation for having the second step. Right? So you can't just take this one and then run Watershed on top and then hope that everything will be like you want it to be. So you need a more sophisticated graph-based agglomeration there. Right? And for this kind of agglomeration, there are a lot of algorithms that can do that. Some are greedy, some are less greedy. So on the gradient, you have the greediest hierarchical clustering. And then on the other hand, you have the globally optimal multicat partitioning. So we evaluated a few. And I think the main question, that's because everyone asks us, is like, well, after you already have this very nice boundary map, can't you just run watershed? And yeah, so we can look. And if you compare those different graph agglomeration strategies, then you see that they are all pretty much equally good, except the watershed, on the difficult data. Right? So here, the errors that we compare, so the A-rand error is like the overall segmentation quality, right? And then in the middle, you have the false merges, right? And then here in C, you have the false splits. So you can see that the DTWS, that's the distance from watershed, right? That's for difficult data, it's really not there. And then if you look more closely, even for easy data, it's, yeah, kind of, okay in terms of false mergers, but as you would expect, it really oversplits it very significantly. Because you would always have this local minima in the boundary landscape, right? And you have these pieces of membrane-like stuff that is inside the cells that can pretend to be like membranes, and they also create more local minima. So in the end, it really oversplits. And that's why we actually recommend, unless you have really, really easy data, to not just use a watershed, but use a more sophisticated graph agglomeration technique. And there are many of them available, and they're all open source, and we have some in our own software, and then there are others. So it's like, it's not hard anymore. Okay, so where we go from here? Yeah, so now you have trained, and now you can see how well it works on unseen confocal data. So this is still the ovules, so it's still morphologically very close to the training data, but this particular data set has not been seen. If we now look at the unseen root data, so which is also, like I said, morphologically close to what we have been before, but a different replicate, yeah, you can see that it's like not doing so badly. I would say it's actually quite okay. But this is actually not far from the data that we've been using in training, right? At least it's the same microscope. It's the same microscope and it's the same model. So what happens if you actually go further away from this? So in this case, let's maybe look at other plant organs. And here we were very happy to find that it generalizes really well. So we think that this comes because of the noise accommodation that we use, and also because we always try to really have, if not exactly the same network, but at least the same technique work for both the light sheet and the confocal data. But at least for other confocal data sets, it really generalizes well. So you can see here is an Arabidopsis meristem from Jan Lohmann's lab. Very different data set, never seen in training, different microscope, still works. Then you see this is not even from our collaboration. It's so George Bassel from University of Warwick has published this wonderful atlas of different plant organs and made it open source. So, and publicly accessible. So we could actually evaluate on all those other plant organs and see how well it's doing. And, there's a lot of variability there. So you can see that like this part, I think it's called anther, is still doing pretty good, right? Even if you go even further away, and then we found this other paper that was also looking at this kind of data, but had really interesting shapes in the cells. So there we were first a bit, afraid that maybe it wouldn't work and wouldn't generalize, because in our ovules or also in the root, the cells are very regular, right? And here you can see that they are more like these puzzle pieces that fit together. So this is the so-called high lobedness cells, so highly lobed cells, something like this. Anyways, it works. Right? So it can follow these weird shapes. It doesn't take shortcuts. It doesn't try to break away, like you can see. I was just seeing some, so you see like it doesn't, for example, try to break this one in two, even though it could look like just two convex cells. And it can also deal with the big variety of cell sizes in the same sample. Like you can see in here, some of the central ones are really, really small, and they're really just that small because in the middle, I guess it's just crowded in there. And then you have the other parts where there's a lot more space, and there the cells are much bigger, and it still works. So we don't have to, like in the, sometimes when you do intensity-based clustering, you really have to carefully select what is your size range and what's the minimum size and what's the maximum size. Here, you don't have to do any of this. It just agglomerates from there. Okay, so it also, of course, works beyond plants, right? So we did everything on plants, but here you can also see the mouse embryo early development from Takashi Hiragi's lab here at EMBL. And there we retrained because the data is really very, very different and the microscope is very different. It still works. So Yeah, then you can also look at this benchmark that we have used. It's the epithelial cells from Drosophila. And there, what we wanted to see is how well would it work even without retraining. So you have this peripodial, I think this one is peripodial and this one is proper disc, but I might be wrong. Don't cite me on that one. One of them is peripodial, the other one is proper disc. And there is training data, but we tried both with and without training. And then just to see how much would retraining actually buy you. So the errors that you can see in here, right, the red ones are the errors. And you see, for example, here, our procedure adds this new cell here, right? Or in here merges the ones in the middle. It happens, yeah. But this is completely without retraining, right? So this was the data set that we just took, and we took our network that we have trained on the Arabidopsis ovules and applied it to this Drosophila epithelial cells. And well, yes, it's not perfect, but you see, it's actually not so bad. So many cells are actually correct. So if you would want to go and correct it manually, there is not that much to do. And the particular gain we can already see. So the peripodial is, I think, the easier data set. And there you see that with no retraining, I don't remember exactly what the measure is that they use. I think it's called seg. But yeah, so with retraining, you can get 10 points better, right? And on the easier one, just 4 points better because it already works better. So you can still gain quite a lot by retraining, but I would dare say that it's usable even without retraining. Okay, so how would you actually retrain if you decided to retrain? Or how would you even start if you had your own data and you decided that you don't want to start from the Arabidopsis ovules because they look completely different and nothing like your data and nothing like your noise. So how would you actually go around it, or how did we do it when we were starting? So you begin with raw data, of course, and then what you need to train any neural network is ground truth images. So you need the images of complete segmentations. And well, over the last year, there have been a lot of move in the computer vision community to go away from fully segmented training data. But I think for this kind of data, in 3D, we still have quite some way to go. So for now, we have to train on fully segmented volumes. It doesn't have to be a complete volume or like a complete root, a complete oval. It can be a 3D subcube, but in this cube, everything needs to be segmented. And really, every pixel has to be assigned. So as I'm sure many of you know, this is very painful and no one likes to do it. And if you just do it from scratch and sit there and paint on membranes, and you will go nuts pretty soon. What we have been doing somewhat successfully is we start from a segmentation procedure that does not need dense labels. In our case, it's of course Ilastik. And yeah, I'll show that to you a bit later. So if you don't know that we are doing Ilastik, I'm telling you that. And in Ilastik, you have a way to segment, which will not be as powerful as the ones that are available by deep learning, but it's much, much faster in the training time, right? Because you can segment just from very sparse labels. So that's exactly what we did. So we took the raw data here, and then we segmented it in Ilastik in the auto context workflow, which provides, I think, the best segmentations you can get without dense annotations. And then out of this, we actually get the preliminary segmentation number 1. Right, so here it is, preliminary segmentation number 1. With that preliminary segmentation, we go to a tool that allows you to correct 3D segmentations. In our case, we took Paintera from Stefan Saalfeld's lab. And yeah, and then someone actually sits there and yeah, you can look at this image. You see there's still quite some things which are wrong. And then you sit there and you correct it. It's also, yeah, not that much fun, but it's not so bad because Paintera tool is really good. So yeah, then you have a corrected image. Then you use that corrected image to train another network that would predict new boundaries. Then out of these new boundaries, you make a new segmentation, you go on, you correct that one again, and then you do a few cycles of this, depending on how difficult the original problem was. And then, yeah, then you do the very, very final polish, and then that one is ready. And then you can use that either for training or for validation later on, because you also need, besides the training data, you also need the data by which you will estimate how well your algorithm is working. And this has to be data that has not been seen in training. So how long it takes really depends on the difficulty of the problem. So in our case, doing the segmentation of the ovals was actually pretty fast because we could jumpstart it from the morphographic segmentations, which were not perfect, but didn't take that long to correct. Now the lateral root data, there was an intern working on that, I think, for a couple of months to create enough training data to actually do all of this, right? Both the validation and the training. Yeah, but then after you have done this, you're all set. Now you're ready. We can train, we can tell you how well it's doing. And also the network that you will have trained by then will be so good that it will only need a few corrections afterwards, and you can then increase the training set on the chip, so to say. Okay, what else is important? So if you're using a pre-trained network, right, like most of you probably will, well, I don't know, maybe you're all network trainers, yeah, more power to you. If you're using a network that was trained by someone else, and I'll show you later on how this can be done, then it's very important that the pixel size, or well, the voxel size, because it's 3D, that the voxel size of this data, of your data, actually matches to the training data that was used for the network training. Because the networks are not invariant to scale. So if it's learned to perform a certain filtering operation, which it encodes in its layers, because it thinks that this is the average width of the boundary, and then you give it different data with a different voxel size, and suddenly the boundary is not 2 pixels thick, but 10 pixels thick, it will be very hard for it to carry over. The problem is you also wouldn't notice, because it would still try to do something, and then you wouldn't even know if it just didn't quite work because the data is too far, or because the network was crap, or but you have to really be careful that the pixel size matches. If you can't make it match, well, too bad, right? Then you do what you can, and then you produce the training data in the way I showed. If you can make it match, that's great. So if you can rescale your own data to the size that was used in the original training, that's cool. Right, so afterwards, remember this agglomeration step that I showed, right? When you're basically have these small bits and pieces like the superpixels, and then out of the superpixels, you build the complete objects. So the procedure that is doing it, where we compared several, but all of them have this parameter that controls if you rather want to under segment or to over segment, right? So if you would rather have to break objects up afterwards, or if you would rather connect the objects out of the existing parts. So you have to choose it by trial and error, right? It's impossible to prescribe ahead of time, because it also depends on the quality of the boundary map, which in turn depends on how close your data is to the training data in the neural network eyes. So we can't really say what it should be, but we provide an easy way to choose this parameter and just verify by eye which one looks better, right? And then finally, once you do this superpixel, so the 3D superpixels are in general better than 2D, but they're much slower. So we did this comparison on this data set that I showed to you with the different parts of the plant, and we tried to see how much would you actually gain if you tune all the parameters that we expose in the software. And here, I mean, that's a lot of numbers. The ones that you should really compare is, so this would be just taking the absolute defaults that we have, the generic confocal network, and then everything else as default. So this would be this line, right? And this would be if you do everything, sorry about that, if you do everything in a nicely tuned way. So this errand error is lower is better, right? So you can see that, for example, for this enter part, it's almost 2 times better. And then for many others, you also get a gain. The only one for which you don't is the leaf. And I think the problem there is rather the quality of the ground truth. Right, so for everything else, it gets much better if you tune the under and over segmentation parameter, and if you really choose the network correctly. Okay. So normally I would ask for questions at this point, but I think we are too many for that. So let's go on. Remember your questions, put them in the chat. I see that there also already been some conversation in the chat there. Yeah. So keep it coming. Think about your questions now, because we are going to, to kind of advance this topic even more. So it would be good if people could understand what's been said so far. Okay. So can we make it even better? Well, the easy way to make it even better is to just annotate more. No one wants to do it, especially not in 3D. So can we make it better without too much annotation? And so here we have developed, or you could say, developed and reapplied the pipeline that we have also been developing for the EM images, of using the top-down priors to break up things which we know are wrong. And the way that works is the following. So recall how the whole pipeline works. You have the boundaries, right? Out of the boundaries, you actually make superpixels, right? So which are like little pieces of objects. And then in this agglomerative step, you actually combine the little pieces into the complete objects. So how does this agglomeration actually happen? Well, I said that it was a graph agglomeration, but to give you a couple more steps, you have the superpixels. Out of the superpixels, you build this so-called region adjacency graph. Right? So basically, you take the superpixel and you take all its neighbors, right? And then its neighbors become also the neighbors in the graph, right? So they are connected. So these are the direct neighbors, right? And these are the indirect neighbors, one step below. And so you have the edges which connect them, and the edges can be positive and negative. So they can want to merge and they can also not want to merge. And then you have the problem where you have the graph and you have some edges that want to merge and some edges that don't want to merge. You don't know how many objects you actually want to segment out. Altogether, this is known as the Multicat Partitioning problem. And it's been applied to images by Björn Andres a long time ago. And then it's been extended to this formulation where you can also consider the not immediate neighbors, also by Björn and Margaret Kuiper, who was then his postdoc in 2015. And then what we have done on top of that is a very simple extension, which was really nice for this kind of a top-down prior introduction. Yeah, so what we do is like this, right? So here you take this pixel, or superpixel in our case, and then imagine that you know that this one should never end up in the same object. Then you can build a sparse edge that separates them, and you can say, okay, these two should never merge. Like, we don't know which of these edges should actually be turned on, and that depends on their own cost and so on. That's why it's a big and NP hard optimization problem. But we just say that we tell it that somehow they should be separated. Okay, and then this kind of sparse edges, if you have the prior knowledge that certain parts of the image should never be connected, that allows you to introduce that into the agglomeration problem and then just solve it. And you can solve it blockwise very efficiently. It's a nice, well-behaved problem. Right, so if you look for an example here, so let's look at this root, right? And I've always been showing it sideways, but it's actually, and remember that's actually 3D. So let's look at the two cross sections the other way. Right, so we have the segmentation here, right? And you can see that this part is, clearly a big false merge. So what prior can we use to say that certain parts of the image should not be merged into the same cell? So of course, you can just say that something should not be too big, but we can actually have a bit more. So I don't know if you noticed when I was showing the raw data of this lateral root, that it doesn't only have the membrane staining, but it also has the nuclei. And these nuclei are somehow biologically interesting. And they are imaged in the same channel as the membranes, right? So we always have them. And usually they really get in the way, because since they are in the same channel, the network needs to learn to ignore them. But in this case, they actually come in very useful, because now the prior that we can use is we can say that we should only have one nucleus per cell. And so we can, you see that they are nicely separated here. The cells are big, so nuclei have space. They don't get merged. You can actually detect all those nuclei here. You see some of them are actually projected from outside, just so that you would know that they are there. Right? And then we can select this two. Right? And we can say that, okay, these guys should actually be separated. And out of that, you will get so you formulate a new problem with this additional edge. And then with this additional edge, you actually get this, you get a different solution, which now looks like this, and it breaks it up in 3. Right? So if you look at what is under there, right, I mean, maybe it should actually be 3, because there is this very faint boundary here, but so deep in the root, it's actually hard to say, because also the multi-view alignment is starting to double the membranes a bit. So yeah, anyway, it's now 2, where it was 1 before. No, it's 3, right? Because this one is also separated, even better. Yeah, another slice. I showed you 2 slices, B and C, right? So this is a different one. We do the same thing, right? So we have these 2 nuclei, right? We say they should be in different cells. We have these 2 nuclei, and then we have these 2 nuclei. And you can see that, so we add these additional edges in there. Now they get separated. It doesn't always work, because if you have absolutely no boundary evidence, like you have here, where these nuclei have oversaturated everything, and then there is just no way you could even cut it there, because it still cuts by boundary evidence. You need superpixels that would allow you to separate it, right? And here, if these guys were all part of one giant superpixel, well, we can't break it up anymore. So yeah, it doesn't fix everything, but it gets much better. So if you have this kind of additional signal and you can think of biological rules that would constrain your segmentation, then this is kind of a general way of how you can apply these top-down rules and constrain your segmentation this way and then get more correct response, even without relabeling anything and without hoping that at some point you will label enough for the network to learn it implicitly. Okay, so we are now through the part where I was just telling you about stuff, and now I wanted to try to show it to you. So yeah, first of all, for the people who want to go the hardcore way and who are ready to just go from our code, of course, all our code is available. Right, so at the top part, you see everything that is for the plants, including all the networks, and everything we have ever done for the plants is there. Right, and there is also the paper and eLife, which I should have cited here, but for some reason didn't, but you can find it. And over there, it's also explained very nicely. If you need very large images, hundreds of gigabytes or something like that, then we also have a pipeline that does it on the cluster. And this is in Konstantin's report. So yeah, that's where you go if you want very large files. If you are brave and want to really go from the code, we are so happy to help you. Just get in touch with us. We are always there. If you're actually not feeling that brave, or maybe not right now, but you first just want to give it a try, we also have other things. Right, so with our collaborators, because we needed to push it to them as soon as possible, because they actually needed to analyze the data and to draw the biological conclusions, we have built a very simple GUI on top, like directly on top of the code, right? This is what we call Plansec. You see, this is really, in terms of how sophisticated GUIs go, this is really just adjusting the parameters of what you would normally pass at the command line. So it has multiple parts here, right? You can put the pre-processing in. It's basically all the stuff that I explained to you in the first part of the talk. You can control all of this here, right? You can select the network, you can select the agglomeration algorithm, you can choose where you dump the data afterwards. So this is, I would say, yeah, still friendlier than just going from code and from the command line, but we are working on this to make it even friendlier. But of course, the friendliest way would be to just have it in Ilastik. And that's also what we are actively working on now. And that's what I want to show it to you. So in Ilastik, if you wanted to implement a pipeline like this, what you would have to do is first, like I said, there are two major parts, right? There's the boundary prediction, and then you have the agglomeration. And we have them separated into two different workflows, where the first one is the neural network classification workflow. You predict the boundaries. And then the second one is the Multicat that actually transforms your boundary prediction into the instances. And then I want to show you how that would work. And for that, I think I need to share a different screen. Let me try this. Okay, so I'll stop sharing that one for a second. See, you're all still with me, I hope. And yeah, let's try on this one. Do you see the Ilastik window?
Yes, we do.
Great. So this is a neural network workflow, which is still in beta, will be out of beta soon because it's getting really nice, I think. And so if you have ever worked with neural networks or wanted to work with neural networks, you probably know that it's much better if you can run them on the GPU, right? So it's much faster. And that's why we actually implemented this kind of a two-tier configuration where you can have the server running on some big machine in your facility, which actually has GPUs there. And then you as a user just connect to it from your client machine. You can also run locally. If you have a machine that has GPUs, you can just start the server locally. That's also no problem. If you don't have GPUs at all, you can still run on the CPU, especially for 2D networks. That's also just still fine, considering you don't even have to do anything, right? You just press predict and it goes. So if we now do that, and let's hope that no one stole my GPUs while I was talking to you. So yeah, you see it's a big server, it has 7. So I'll just take this one. And then we go off and we take some training data. Not training data, sorry. We are not training, we are just applying, okay? So here, what I want to show to you is how you can take a pre-trained network. And like I said, all the work that we have done for the plants, we have made them all available. So you can download them and, for example, try them out here. Right, so we can, let's see, can I make this a bit bigger? Yes, okay, so there you see part of a route that is very different from the other routes I was speaking about. That's from the plant atlas, never seen in training, totally new data set. Suppose that you actually came to us with this data set and you want to try it out. And I'll show it in 2D just because it's faster this way. So this is your data set, right? And then you actually go to the neural network selection, and I can't do it because I have my screen sharing thing there. Okay, so then we load the model. So this is ours. It's from Arabidopsis ovals 2D. So we say open. What it does now is it downloads the weights of the model from, because we don't distribute them right away, it just does it on the fly. Okay, so the network is loaded, and then we can just say predict and see what happens. Right, so now what it does, it's sending the request to the server, who is actually predicting the boundaries. And here you can see the prediction for, I'll make it a bit fatter. Right, so you see the prediction for the boundary label. Right, and it's coming, it's coming. So the computation is actually very fast, but it also takes a while to carry the images back. Yeah, and you see the prediction, considering it has never seen this data before, I would say is really not so bad. So we have the boundaries, and I think that was kind of the easy step. What you can also do is you can select several networks here and just try them out sort of one by one. Especially if you're not sure about which one would fit better, that's also a good way to try. You can, of course, load more data sets. You can stop the prediction here and now then do the data export there and actually export your predictions. So I think that is all supported. So we will. So it's in beta now, and there are some trusted users trying it out now. Well, if you want to become such a trusted user, please get in touch. We are very happy to help. And we will make it out of beta sometime soon too. Okay, so let me stop sharing this one. So I hope you have all seen how to predict boundaries now. If you already have a pre-trained network, let me show you the other step, which is actually agglomerating from the boundaries then. All right, so for that, I'll need another window again. So I'll stop sharing now. Hello again. And let me maybe close this one. I'll close this Ilastik. And I'll open another one, and I'll share with you the other one. So now the workflow that we need is called boundary-based segmentation with MultiCAD. And I'm ready to share again. You see the Ilastik window?
Yes, we do.
Very good. Okay, let's load some data then. And we will, of course, load the data that we have processed before, right? So we have the raw data of this root, and we have the probability map that we have just now predicted with the neural network. Right, so that's the one. Right, and you see that's what we have seen before, just in different color. Right, and then we can try to break it up into superpixels. Right, you see that would happen. There are some parameters to control there, but they are actually quite visual, I think. So here you can make more of this edges count, right? So you go down, now that's too much. Okay, 10 is probably, yeah, we can, for example, keep it that. Now let's look at the superpixels. What you have to watch out in the superpixels is that things that you think might be separated, but the boundary signal is there is very low. You want to have two different superpixels there, because otherwise you don't even have a chance of ever separating them. Right, so supposing we were happy here, so then we just go there. Right, these are our superpixel boundaries. Maybe I'll make it a little bit bigger and zoom out a little. Okay, now we see almost the complete thing. And yeah, so we go there and we update, right? And now, yeah, so that would be your segmentation. And we can look at the whole segmentation too, right? So that would be the segmentation. And we can also change the color just to, yeah, I think that's probably better. And then we can say, well, okay, we had this other parameter. Remember I told you you can control the over and under segmentation, right? That you can see if you want to break it up more or less, right? So this is this beta, right? So we can say 0.6. Let's see what happens now, right? You see other things, they start appearing, right? We can say 0.7, right? And now also more of them get cut out. You can also go the other way, right? Well, we can even say 0.8, right? See what happens now. That's probably too much, right? You see it start breaking things. So you play with that. And then again, once this one parameter is fixed, it's ready to go for as many data sets as you want. And then you have the data export, you have the batch processing. So you can go on from there. Okay, so if you have questions to that part, write them in the chat. I'm not going away. We will clear it all up. let me go back to the presentation sharing. I wonder, can I just, new share, look at this. I can just switch like that. Huh. Oh, I didn't even need to stop before. Okay, so just to recap on that, and that's the slides that I have kept in there in case the demo wouldn't have worked. We have the neural network workflow that can predict the boundaries. We have the superpixels, then you start, so you save all your boundaries, go to the Multicat workflow that makes boundaries into objects, break it up into superpixels, and then you agglomerate into your final root. And then you just export that. So it's still more steps that we would want to have and more parameters that we would want people to get confused in, but we are working on it. We hope to make it friendlier. Our collaborators are actually very happy with the whole segmentation quality and are ready to jump through a few hoops that we haven't removed yet, but overall we are working on making it as friendly as we can. Okay, what if you need a different network? Right, so we have trained on our plants in confocal and in Nightshade, but what if you actually have something like none of them really fit and all the boundary predictions are not so good? So what you can do is, well, remember you need training data. So if you had training data, this Plansec, the simple thing that I showed you in the few slides back, it actually supports command line retraining. So we would keep all the hyperparameters that we have found to be good for our training, and then we just put in your training data, it trains. If you don't have training data, or if you don't want to bother with all that stuff, or if you don't have GPUs, right? For training, you absolutely need GPUs. If you just want to predict, you can get away with just CPUs. For training, it's absolutely indispensable. If you don't have any of that, or if you just want to see what the others are doing, maybe there is a different network, well, then you go to the network model zoo. And that is the model zoo. So you see there are a lot of things in there, and we've been adding more. This is a little community effort for now, which is shared between us and ImJoy and Fiji and DeepImageJ. And the zero cost GL guys have also kind of on the verge of joining or almost joined. And so this part is, for now, very much, I would say, developer centric, because we first wanted to make it good for ourselves to put models in there. But now that we are past this part and we have put in a lot of models, and I think we have fixed the format, our next target for November is to actually make it friendly for the people who just come there and look for their own models. So that's the place where you will go if you find that the networks that we have provided are not good enough, or actually if you even just want to see the networks we provide, they are there, right? So that's where we distribute them from. So yeah, that's a website to watch, maybe not for tomorrow, not for Saturday, but 2 weeks from now, a month from now, that's where you would go. And we will, of course, announce the launch everywhere and on Twitter. So once you see it launched, you go there, it will be all very nice and clear. If you actually train networks yourself and want to share it with people, get in touch. We'll tell you how to do it through this thing. We'll hold your hand. We'll try to be as much help as we can. To summarize, if you need to do boundary-based segmentation, that for now, the most reliable and robust workflow is to do boundary prediction first and then do the agglomeration from the boundary prediction. We have seen that generic pre-trained networks actually work well and generalize well to unseen data. Well, it's already available in our Plansec tool and of course in all our code, and it's a beta in Ilastik. And so we hope to improve it even more and push it out soon. To finish by thanking the people who've also been part of this work. So we have wonderful collaborators that provide the plant data and that make sure that we actually create something useful. They're also the great guys who work on the bioimage.io website and on the whole model zoo. And this is my group in our socially distant group retreat this year. Thank you very much for your attention.
Thank you very much for a fantastic talk, especially with the live demo. That was super cool to see how this works in practice.